Yeah, thanks, Matt. This is a topic that I think Matt and I probably think an outsized amount about. And I think it's something that's getting to be really, really important, particularly for our breed associations as these low pass genome sequencing products come on the market. How do we maximize the effectiveness of that imputation so that that downstream product is every bit as reliable as maybe what we would have gotten on a chip, right? So again, I think that we need to be intentional about this, and my hope is that we can set up sort of a vision for what that might look like. I'll probably be quick on this talk because I'd like to stimulate a little bit of conversation amongst breed association folks, folks at US Mark that I know are thinking a lot about this. So with that, I'm going to jump right into things here. And really the low pass sequencing and imputation, this is coming, right? And I'm not going to forecast when it's going to overtake chips, but the reality is this is I guess nearly an 8-year-old figure of the cost of the genome dropping at a rate faster than Moore's law, right? So how fast can we make computer chips smaller and better, right? We've done that and more with decreasing sequencing costs, okay? And this is only going to keep getting cheaper. There are multiple sequencing devices on the market that have added a little bit of competition to this marketplace, which is good. And I think that these costs are going to keep going down in the technology that we've got to utilize these sequencing resources in a genetic prediction context are really increasing. The big advantages, and we'll keep coming back to this idea of rare variation. As a researcher, I'm very interested in the rare stuff, right? The things that don't show up on that 50K chip of very common variants that we've sort of set aside for our genomic prediction reasons. But also some flexibility with low pass, not to have to go back and redesign chips for SNP discovery, for detecting CNVs and structural variation. I think all of a sudden we unlock a new set of questions that we can ask and answer and implement into genetic evaluation through the utilization of low pass sequencing. And so when we talk about low pass or low coverage sequencing, just to get everybody acquainted, coverage is the average number of reads that we have at a particular spot in the genome, right? And so keep in mind that sequencing is completely random. We bust the genome up into a bunch of pieces. We read across those bunch of pieces, and then we try and put everything back together in a logical way, right? And so the important thing to realize in the context of low pass sequencing in particular is that coverage is calculated genome-wide and not necessarily on a site-by-site basis, okay? So there are going to be sites in the genome that get covered by, hundreds of reads, right? And there's going to be sites in the genome, even in a high coverage, say a 10, 15, 20, 30x genome that might only get a couple of reads touching them, right? This is sort of the toy example in the picture here. So, again, in low pass, this is just much sparser, right? Much of the genome is not even getting touched by a single read. There might be some places where we're fairly confident, for example, there where we get 3 reads maybe that overlap a particular section of the genome there. And our challenge, right, is to take this information, this low density, low number of reads, and turn it into something that's representative of genome-wide genotypes, right? And so this is a big, complicated set of, this is the Glimpse software, sort of the industry standard for doing low pass sequencing imputation, right? So we start off, of course, over here. I don't know how the clicker works, but over here with genotype likelihoods on the far left. So again, sparse reads across the genome. Some of these get covered a handful of times, and we get some pretty high confidence calls on genotypes. Others flip a coin. We don't have any idea about it. But the key in what gets us to this other side, where we have a usable product in terms of a genome representative imputed genome-wide sequence, is this haplotype reference panel. Okay. And so really the focus of my talk is, how do we optimize and select the correct individuals that reside in this haplotype panel such that our imputation is maximally accurate? And again, just before I dive into strategies and the moving pieces that make this happen, I think it's helpful to visualize what imputation looks like from a resolution standpoint. This is from a genome-wide association study that we did on one of my PhD projects, looking at the same peak, the same individuals utilizing SNP50 genotypes, right? So these are just pulled off from the typical SNP50 assay. Then down to a 850K imputed set of SNPs, so much higher density, but still very far from sequence imputed. And so what we find when we've only got 50,000 SNPs, these represent big LD blocks, right? They're good enough for us to do genomic prediction, but our ability to go in and map causal variation or identify those variants that are in perfect LD with the causal variant is virtually zero with a SNP50. Even with the 850K, some of these sub-associations in this complex GWAS peak, we're not able to pick those up, right? But then we get into the sequence imputed density, and we have this really fine resolution to look at those variants and get into the functional biology, perhaps of a GWAS signature like this. And I'll come back to this image at the end with sort of a charge for the animal breeding community. So what does accurate imputation need? I think it really needs three things. My text box got a little bit messed up here, but we need a large reference set of haplotypes. And that's going to be the focus of what I talk about. Is we have to have a set of haplotypes where we're very confident in the genotypes and in their phase, which chromosome they come from, that are representative of the target population. This is just like calculating a molecular breeding value, right? If we don't have the target population represented in the reference, we're going to do a poor job of generating imputed genotypes for these individuals. We need a high quality reference genome, which in cattle, luckily we've got. And then a recombination map, understanding how recombinations happen across the genome helps us more accurately fill in the blanks when it comes to imputation. And so the big challenge with imputation is we're very good at imputing common variants, but it turns out that most of the variation in the genome is quite rare. So these are actually three separate assays and their relative minor allele frequencies. You'll see the SNP50 in yellow there is overwhelmingly common variation. That was by design. We picked these evenly spaced markers that were high minor allele frequency in the population, and that's contributed to our ability to do genomic prediction, right? But the reality is that the actual minor allele frequency spectrum of the genomes of populations of cattle looks a lot more like that F250, where we have an overwhelming amount of those rare variants in the 0.01 to 0.05-ish range. And the vast majority of the variants are going to be in that rare part of the minor allele frequency spectrum. The challenge of imputation is it's really just pattern matching, right? We see a haplotype in the reference population, we fill in the missing pieces in this, whether it's low coverage sequencing, whether it's a genotype array, whatever that is, right? We want to fill that missing stuff in. It's got to be represented in the reference population. If it's not there, we do a much poorer job of imputation. And so when we look at, this is just looking across the minor allele frequency spectrum and how we do with imputation. So imputation quality score in this paper was our quantification of how well a site was imputed. And you'll see that there's a massive fall off once we get to about 10% of the minor allele frequency. Those things, our imputation quality drops off pretty fast. And so rare variation is our challenge. How do we represent it and find the most representative set of haplotypes such that we can maximize the effectiveness of imputation? Again, whether it's coming from existing chips that live in our database, whether it's coming from low coverage genomes, making sure that we're representing the haplotypes of our current population as best as possible. And so the big questions that I think Matt posed to me is one, who do we sequence? And I'll spend most of the time thinking about that. But then there's also some caveats. Just because we sequenced animals doesn't mean that the data is ready to rock and roll in an imputation pipeline. We need to do some evaluation of how deep we sequence these things and how we can intentionally go through and update both our reference population as well as imputed samples in the genetic evaluations as we get down the road here. And so before we even talk about sequencing for imputation, I think that the previous talk was a perfect lead into this. My argument is that every AI sire should probably be sequenced at this point. When it comes to a genetic defect popping up in a population, our ability to go back and work through that really, really fast is a lot easier when all of these animals have a high coverage genome, right? Sequencing has gone down in cost. It's no longer a $5,000 or $10,000 endeavor to get a high coverage sequence. We're talking, in the $500 to $700 on the upper end of a reasonable coverage genome. So I think that this is a way for us to not only build a quality imputation reference, but it's an insurance policy. It allows us from a research standpoint at breed associations or wherever to be proactively monitoring de novo mutations, lethal haplotype mapping at a resolution that's just not possible with, say, 50K SNP data. And again, this is always going to feed back into an imputation pipeline that allows us to maximize the utility of those low coverage genotypes that we're able to generate. And so how do we build a reference panel? The first thing that I'll sort of dig into is doing a single breed specific reference versus a multi-breed imputation reference and what that sort of looks like. We have done some work. This is from, again, a paper that we wrote back in 2019, focused on imputation from low density SNP chips up to about 900,000 SNPs. So again, not a one-to-one comparison, but I think that these patterns, particularly as it relates to the multi-breed versus the single breed reference panels, are going to hold when we move into either sequence imputation from chips or into the low pass world. So admix populations in particular are going to benefit from borrowing haplotype information from animals that aren't labeled as their same breed, right? So if we've got a population of Sim Angus animals, right, having both Simmental and Angus in that haplotype reference is really, really important. But also all of the other admix breeds that maybe have some Angus haplotypes floating around, we're always better off having those individuals in our reference. And again, label population not equal to actual population. And there's a lot of haplotypic diversity that's been passed along to all of these between these breeds, right? That has certainly muddied the water, such that we can't just grab a pool of animals that are labeled X and expect that to be the optimal way to impute another set of animals labeled X, right? And the other thing to keep in mind about imputation is it really is a pattern matching strategy. It's typically not going to impute stuff in that if there is the correct haplotype that lives in the reference, it's not going to impute something incorrectly, right? In all the work that we've done, we've never hurt ourselves by having a larger, more diverse reference panel. So I would argue, particularly down here in the rare minor allele frequency side of the spectrum, the likelihood that you're going to see a haplotype represented in that big multi-breed reference is going to increase those accuracies, and that's exactly what we saw at the level of high density SNP chip panel imputation here. And the other thing too, right, when we think about pattern matching and in the context of an open herd book, so this is using some Gelvi genotypes with both a breed specific reference. So these are all animals that are labeled Gelvi versus a composite reference of all of the genotypes that we have access to. What you'll see is that the animals that get imputed well with the breed reference, with the breed specific reference, they don't see any change in their imputation when we move over to a composite reference. But these animals that get poorly imputed in a breed-specific reference, these are likely admixed animals, balancer bulls maybe, right? Once they can borrow additional information from that multi-breed reference, we do a much better job of imputing them. And the big kicker, oh, my table didn't show up there on my white background. If you could read white on white, what you'd see is that if you have a population from a breed standpoint that's adequately represented in the reference, the imputation quality is really impeccable, right? So this is again from 50K up to about 900,000 SNPs. We're 0.99 R squared in any breed that has more than about 100 individuals represented in that high density reference panel. So again, we do a really good job of imputing animals as long as their relatives or animals from their breed are well represented in the reference panel. And I think that this idea of ascertainment bias and what a reference panel looks like, particularly in the context of rare variation, is important. So This is a human example where compare a couple of coverages of low pass sequencing and imputation, as well as an Illumina chip genotype. And we've got Europeans and African populations, right? And so in European populations where these chips were designed, again, this ascertainment bias idea, we design a chip for this population. It does a pretty good job, the chip does, of imputation. But when we move over and look at that chip genotyping an African population, again, not the population in which the chip is designed, we see a pretty substantial drop off in imputation quality there. So something to keep in mind from a cattle context as well. And this is sort of where I start moving into recommendations as we think about which animals get sequenced. So this is from a project we've been working on with the American Simmental Association, identifying sires that would merit being sequenced for an imputation reference down the road. And so the big red blob here, each one of those tiny sort of transparent dots is an individual, a principal component analysis that helps us sort of visualize the population structure here. These blue dots in the middle are our top 150 sires from which we were looking to sequence for animals that'll end up in our haplotype reference panel. My point here is that if we just focus our sequencing efforts on overrepresented sires as far as registrations, we're probably being quite redundant, right? We're sequencing the same haplotypes, the same generally closely related individuals, but we're missing out on a lot of haplotypic and relationship differences that exist across this whole population, right? So I guess my sort of recommendation is that we could do a better job, or we'd be better served not looking backwards, reaching in and finding legacy sires. I think there's a risk there when you go and sample a bunch of old haplotypes, you try and impute those, and all of a sudden those haplotypes have had maybe 50 years of recombinations to break them up, right? I think that our interest should be in sequencing as many haplotypes that exist in the population right now. And I think that we can use our chip genotypes to do this, right? So whether that's on an individual level with some sort of a clustering algorithm, whether that's going through and doing an actual haplotype search, saying this is a very frequent haplotype that we don't have in our sequenced population, and then condition a subsequent round of haplotype search on that individual being sequenced to find a targeted set of animals that have haplotypes that are going to help out this imputation reference downstream. So there's lots of ways to do this, and I'd be happy to bounce some ideas off of folks in the room once I wrap up here about what this might look like in practice. And just a couple more things to keep in mind more than anything. How deep do we sequence? This is something that I attribute to one of my former mentors, Dr. Bob Schnabel at Mizzou. Bob always said garbage in, garbage out. And I think that this is very true for imputation references as well. If we put garbage sequence into an imputation reference, we can't expect it to impute low pass genotypes or chip genotypes well on the other side of the little algorithm here. So again, Bob and I have been doing a bunch of work on the quality of these reference genomes that go into the imputation reference, both from a sequencing depth standpoint, seeing where we need to be from a sequencing depth standpoint, as well as from optimizing phasing algorithms to make sure that we're getting actual haplotypes together from the paternally and maternally inherited chromosomes. So this is a representation of chip genotypes and genotypes from sequence. So these are animals that are both sequenced at high coverage or at various coverages from about 5x all the way up to nearly 40x, and their chip genotypes, right? So this is assuming that chip genotypes are the truth set, and we're asking what's the discordance rate between the chip and sequence genotypes. And you can see there's a really clear plateau down here. Once we get to about 10 or 15x, the number of discordances that we see between SNP chip and sequence genotypes really starts to fall off, right? So my recommendation from a sequencing depth standpoint, the additional cost, again, with a little bit of competition in our marketplace here between Element and Illumina are the two that come to mind immediately to me. If we can sequence another gigabase for, somewhere between $2 and $5, The marginal increase in cost between a 5X genome that lives in this reference population and a 15X genome is 60 to 150 bucks, but we're so much more confident in those genotype calls, right? This is, again, the garbage in, garbage out situation. We want to make sure that the genomes that we put in are as good as possible. We see a similar thing here in the context of Mendelian errors. So these are duos. So you've got a parent and an offspring sequenced, just looking on chromosome 25. And you can see that in this 10 to 20x and particularly over 20x sequencing depth, we see a substantially lower proportion of these Mendelian errors, indicating that those genotype calls are really quality and as good as they can be. We don't probably get a whole lot of additional value out of that 30, 40, or 50x genome sequencing. And then the last thing that I'll talk about is that phasing matters too. It's one thing to sequence, but taking those genotypes and turning them into haplotypes that live in this reference is also a really important piece. The good thing is that we have very deep pedigrees. These animals are all quite related to one another. So again, our pedigrees can do a big proportion of the phasing before we even have to enter it into some sort of an HMM algorithm. Shape-it5 is the one that we've been playing around with most here, to go from that unphased genotype into a useful haplotype at the end of the day, as well as utilizing read-back phasing. We can resolve haplotypes just based on the contiguous sequencing reads. And again, that just helps out the quality of that haplotype that lives in the reference. And the final piece that I'll sort of leave us with is that if we want to do this imputation, I think it also requires that we evaluate accuracy regularly. We understand the impacts of adding new animals into the reference, changing the reference, and that will feed back into this idea of how often do we need to update our routine genotypes? Do we need to re-impute these? Is there that big of an accuracy difference? So again, my encouragement is anyone who is getting into this space, finding a set of testing individuals that have high coverage genomes, that you can downsample to low coverage or to chip genotypes, and then ask how good is the imputation, both on a per SNP basis. So finding places where, regardless of how many animals we have in the reference, regardless of how well we represent these haplotypes, some of these SNPs or small variations just don't impute well at all. We do well not to utilize those downstream in genetic analyses. And then finally, on a per individual basis, is there something structural when we see this little blue animal that imputed terribly compared to all the rest of his contemporaries? Can we ask why, and then use that to inform a sequencing strategy, or how we maybe handle that data in order to maximize imputation quality across individuals? And then finally, pangenomes are coming. How do we utilize those in a breeding context? I really don't know. There's a lot of questions surrounding, maybe in 10 years, a 10x genome on everything is possible, but how do we store that? How do we move that around? There's some big structural questions that in the genetic evaluation standpoint about how to handle data like this that I think bear some more conversations. And then finally, how do we utilize this additional variation to improve predictions? The jury remains sort of out on, are these informative sequence variants actually useful in increasing the accuracy of our genomic predictions? And I think that the genomic prediction workshop in December is going to cover this in some serious depth. So I hope that this is just spurring a little bit of thought in the lead up to that as we try and tackle what having all this data at our fingertips means from a genomic prediction perspective. So with that, there's some takeaways, my contact information, and I'm happy to answer questions from anybody and everybody for the next couple minutes here.